I'm Gillian Peterson, Associate Professor at the Centre for Microbiology and Environmental Systems Science. I study symbioses, which are interactions between animals or plants and beneficial microbes.
Hi, and I'm Michael Wagner. I'm a colleague of Gillian's at our center, and one of my research interests is to study the function of microbes directly in their habitat. Today, we will show you how antibiotics were discovered, how they function, why antibiotic resistance is such a huge problem for humanity, and why we must act now to ensure that we will still have these life-saving treatments in the future. So we are doing this video together because we have complementary expertise in microbiome research.
I study host microbe interactions and fundamental aspects of how they interact. And this is really what's happening when an antibiotic-resistant pathogen infects a human or an animal host.
And I am interested in the function of microbes in complex communities as they occur in the human gut or in wastewater treatment systems or in rivers. And that's where also these antibiotic resistance genes are exchanged between microbes.
You could finish school in Austria or in Australia, where I'm originally from, and never learn about microbes. The first thing taught in the school biology classes I know is trees, which is strange because usually you start at the beginning. And for life on planet Earth, microbes were the beginning. Microbes are all biological entities such as viruses, bacteria, archaea, protists, and fungi that are too small to see without a microscope. They were the first organisms to evolve around 3.8 billion years ago. And today, they make up most of the species living on this planet. There are more than 1 trillion bacterial species on Earth, but only 1 in 100,000 has been well studied. They colonize every imaginable habitat, including our own bodies, which contain more bacteria than human cells. Microbes are incredibly important. Without them, we would quickly run out of oxygen in the atmosphere. The plants in our fields would die due to lack of nutrients, and we could not digest a single meal. Most microbes are beautiful and fascinating and diverse and helpful and important for human and planetary health. However, there are a few bad apples, a very small subset of species that cause disease. You might have heard some examples. Salmonella that causes diarrhea, Staphylococcus that causes skin and wound infections, and others. Today, most of us don't think too much about bacterial infections. We have a wonderful weapon against these, antibiotics, which all of you have probably taken in your lives, likely several times. Before the discovery and widespread use of antibiotics to treat bacterial infections, the average human life expectancy was 46.5 years. Afterwards, thanks to this and other medical breakthroughs, it's 72.8 years. Antibiotics have saved millions of human lives so far.
Jill mentioned that microbes are incredibly diverse. For example, pick up a teaspoon of soil in your garden. It contains tens of thousands of different microbial species, all competing for space and resources. One of the ways that microbes evolved to be competitive is to inhibit or kill one another with chemical weapons, called antibiotics. Today we know that many microbes produce antibiotics. But how did we first discover these compounds? The story begins in 1928, when the Scottish microbiologist Dr. Alexander Fleming returned to his rather disorganized lab after a weekend vacation to find something unexpected on the petri dishes he was using to cultivate Staphylococcus bacteria. A type of blue-green mold had started growing on his experimental plates. What caught his attention was that the bacteria around the mold weren't growing. After further investigation, Fleming realized that the mold was producing a chemical that could kill bacteria. He named the substance penicillin, the world's first discovery of an antibiotic. The compound was later purified by other scientists, but the process was cumbersome. In 1941, the United States still did not have large enough penicillin in stock to treat a single patient. However, the large number of deaths from the bacterial infections during World War II sparked a major effort to improve this process. And by September 1943, enough penicillin was produced to satisfy the demands of the entire Allied armed forces. No fewer than 21 US companies joined forces to produce 2.3 million doses in preparation for the D-Day invasion. And penicillin quickly became known as a miracle drug, saving at least 300,000 lives during World War II. As penicillin was not effective against all pathogenic bacteria, a broad screening of cultured microbes for new antibiotics was initiated, which led to the discovery of soil-dwelling actinomycetes, for example, as prolific producers of antimicrobial compounds. During the golden age of antibiotic discovery from the 1940s to the 1960s, many new antibiotic classes were found that are still in use today. In fact, the rapid and relatively easy discovery of multiple classes of antibiotics during a relatively short period led to the excessive use of these drugs outside the treatment of life-threatening diseases. So how do these antibiotics work? They work by targeting essential processes in bacteria, either stopping their growth or killing them directly. As human cells are biochemically different, they are generally not affected by antibiotics. But what is now antimicrobial resistance, and why is this a crisis of global concern? Antimicrobial resistance occurs when bacteria develop defenses against the antibiotics, designed to kill them. A bacterial enzyme that could inactivate penicillin was already discovered in 1940. The microbes able to make this enzyme are resistant to penicillin. Penicillin resistance was also detected in hospitalized patients infected with Staphylococcus aureus, a pathogen. And with increasing use of antibiotics, by the late 60s, more than 80% of Staphylococcus aureus strains found in patients were penicillin resistant. Unfortunately, the emergence and spread of antibiotic-resistant pathogen strains was not confined to penicillin, but has been recognized for all newly introduced antibiotics, eroding their effectiveness for the treatment of infectious diseases. Another mechanism of resistance is to alter the specific proteins that the antibiotics usually bind to. Bacteria can also prevent antibiotics from reaching their target by increasing efflux pumps, which actively remove the antibiotic from the cell. When antibiotics are used to treat an infection, they kill the susceptible bacteria, but bacteria with resistance genes survive. These resistant bacteria can now multiply without competition from the killed non-resistant bacteria. Over time, the selective killing creates an environment where the resistant bacteria have a growth advantage, leading to an increase in the overall population of resistant strains. Overuse or misuse of antibiotics accelerates the evolution of resistant bacteria by constantly selecting for resistant types. Unfortunately, once evolved, resistant genes can be shared between bacteria via plasmids or other genetic elements through a process called horizontal gene transfer. Due to the evolution of antimicrobial resistance, there are now pathogenic superbugs that are resistant to all known antibiotics. As a result, we could fall back into a pre-antibiotic era in which minor injuries or infections that were previously considered trivial could once again be fatal. Medical procedures such as cesarean sections or hip replacements would carry a much higher risk. There is therefore an urgent need for new classes of antibiotics. Unfortunately, however, the traditional antibiotic discovery pipeline is running dry, as almost all antibiotics that can be produced by easy-to-culture microorganisms under laboratory conditions have already been found. Antimicrobial resistance is one of the top global public health and development threats, causing 1.27 million deaths in 2019. As bacteria evolve and become further resistant to existing antibiotics, the challenge will only grow. Some estimates suggest that without reversing this trend, antimicrobial resistance could lead to 10 million deaths a year by 2050. This is a silent tsunami, disproportionately affecting the most vulnerable. 1 in 5 deaths from resistant bacteria occurs in children under 5, and the burden of antibiotic resistance falls disproportionately on low and middle income countries.
Despite what you've just heard about antibiotic resistance, we humans are still making the problem worse. Every year, thousands of tons of antibiotics are used on farms, meaning they're given en masse, like food, to otherwise healthy animals because, for reasons not fully understood, they make them grow faster, improving yields. They're also used prophylactically to prevent infections when farm animals are kept at very high densities under poor conditions that promote stress and infections, as for example in battery cages or aquaculture ponds. The overuse and misuse of antibiotics in farming contaminates global environments with resistance and is one of the major reasons behind the rise in untreatable antibiotic-resistant infections in humans. So what can we do to prevent the tide turning back in favour of the pathogens? Firstly, we need more research. With new vaccines, for example, we could prevent infections before they require antibiotic treatment. We need to better understand how resistance mechanisms work and how they evolved. Such knowledge recently allowed researchers to re-engineer old antibiotics to restore and even enhance their effectiveness. But we will also need new classes of antibiotics to stay ahead of resistance. For this, we could learn from nature. I mentioned that only 1 in 100,000 bacterial species have been studied in detail. The rest represent an enormous biobank of new genes, molecules, and functions with commercial and medical applications we now can't even imagine. At our centre, we're developing methods to study them, even though most we can't grow in our laboratory flasks. Out in nature, bacteria can also be infected and killed by viruses called bacteriophages. Scientists are working to harness bacteriophages as novel treatments for antibiotic-resistant pathogens. But this is challenging because bacteria can also evolve resistance to these. In addition to more research, there is something we can all do right now. We must use the antibiotics we have with extreme caution. They should only be prescribed in humans when absolutely necessary, not for minor illnesses that we would recover from by ourselves, and definitely not for common colds caused by viruses. Viruses are completely unaffected by antibiotics. We must also stop using antibiotics for growth promotion in farming. This practice has been banned in the EU and UK since 2006, but it must be a global effort because it contaminates our food and our natural environments with resistance genes that can spread rapidly around the world. We all need to eat less meat, and we need to pay higher prices for meat that is produced under humane conditions where antibiotic overuse is unnecessary. Thank you for listening today, and if you'd like to find out more about microbiomes, come and visit the website of our centre.